Dear all, I'm very happy to see you, and I'm very happy to present our today speaker, Mário Mendes, who will talk us about early angiosperm from Portugal and about the surrounding vegetation. Well, Mário, you may start.
Yes, let me put this. Can you see?
Yeah, full screen is okay.
Oh, okay. So, good morning, good afternoon, or good evening to everybody, because I noticed that there are people from several parts of the world here. And Natalia, I'd like to thank you for this nice invitation to talk about the early Cretaceous vegetation and the emergence of angiosperms in Portugal. As you can imagine, 20 years walking with the early Cretaceous floras from Portugal is too much, and I select some parts. I cannot, it's impossible to show you everything. And I select the most important things for me about the early Cretaceous floras. This has been a very long journey since 2006, and there are too much to talk. Well, let me check where is the point. Yes, yeah. Uh, our studies have been developed here in the Lusitanian Basin, very famous in the world. This basin is very important and was formed during the Atlantic opening. Is a very big extension, about 200, 300 kilometers, and is very important because here in this basin, located in the west part of Portugal and is the westernmost Iberia, we have all the Cretaceous very well represented since the Berriasian until the Maastrichtian. So we can follow the evolution of the vegetation since the early Cretaceous, dominated by ferns and gymnosperms, until the late Cretaceous, dominated by angiosperms. Of course, the presence of this basin sparked the interest of many people to a long time ago. I select two, Saporta and Carlos Teixeiras. There are many others, like Oswald Hare, Venceslau de Lima, and others. But these two persons were very important to start the studies about the angiosperms here in Portugal. The classic cliff floras from the Cretaceous of the Lusitanian Basin was studied from Saporta and published in 1894, and he reported the occurrence of angiosperm remains in several places, but I select two ones, the most important and very known and several times appear in the literature are the floras from Cercal and the flora from Buarcos, Tavareda. There is another work, more or less contemporaneous from the Saporta studies, but developed in the other side of the Atlantic Ocean. I refer to the Fontaine works from 1889. Is a little later from the Saporta works was a little later from the Fontaine studies carried out in the Potomac group deposits. The works developed by Saporta and Fontaine was important because they have influenced many people on the study of angiosperms from the early Cretaceous floras in several parts of the world. Here in Portugal, we have Portuguese, very famous here, Carlos Teixeira. And Carlos Teixeira revised the floras described by Saporta between 1945 and 1952. These studies were important because provided a general overview of the vegetation in Portugal during the Cretaceous period. I will talk only about the early Cretaceous, but Carlos Teixeira's study also the late Cretaceous floras. And of course, based on these studies, um, we can they prove that there has been changes or turnover or whatever among the floras of the early Cretaceous, dominated by ferns and gymnosperms, and those one from the late Cretaceous, largely dominated by flowering plants. These researchers have study leaf fossils, and we have a problem with these fossils because many of them were preserved as impressions, and as you know, it's very difficult to extract material to prepare cuticles, for example, and the information that can be obtained from these specimens is a little limited. Well, here I have 2 pages from the book of Saporta, Flor Fossil du Portugal, and I'd like to show you this specimen here, described from Gaston de Saporta as Schopfatia franchetti. This specimen was collected in Cercal flora, and according to Saporta, is an angiosperm. I never saw the specimen. I never have the specimen in my hand. I only see the drawing, and we must be some careful with the support drawings because he had a great imagination, and sometimes the drawings was a little exaggerated. But according to Saporta, Schofatia has a main axis with long roots. Later, Carlos Teixeira observed the specimen, and he preferred to not make additional considerations because for him the specimens is not clear, and he only said that the specimens yield very thin leaves, as you can see here, and probably was a floating plant. However, he did not say that the specimens was not an angiosperm, but of course he was not certain. Here we have another interesting specimen, also from the Cercal flora. These leaves, these fan-shaped leaves, very nice. I have observed these fossils because there are several specimens in the Geological Museum of Lisbon, and Saporta described this specimen as an angiosperm leaf, but in my opinion, is not an angiosperm. He described it as Protorhipis schofati. The specimens was collected from Cercal flora, and these specimens are preserved as impressions. In my opinion, this is not an angiosperm, and I will explain why. If you look careful at this picture here, you can see a reticulate venation. I cannot see free veins here, and I did not see also a main vein as is usually in angiosperms. Besides, there is another factor, important factor. Here you can see these scars, these rounded scars, spread on the limbals, and in my opinion, this is an aquatic fern because these scars, I'm sure that are sporangia. Later, Carlos Teixeira observed the Protorripes shafati described by Saporta from Cercal, but he changed the genus. He transferred the specimen from the Protorripes genus to the Nympheites genus. So this species suggests an aquatic habit and its attribution to angiosperm, but is uncertain. The circle leaves, as I've told you, have reticulate venation, which is different from angiosperms. There are no free veins, and the small and round scars on the limbals may correspond to sporangia. There are also some marks in the leaf margin interpreted as vegetative buds. So I feel that this specimen, Protorripis shaffatia and later Nympheites, may correspond to the extant Ceratopteris cornuta or Ceratopteris pteridoid with sporangia scattered along the veins of the leaf with vegetative spots here on the leaf margin. Here we have more specimens from the early Cretaceous floras, but from a different flora. This is more important to the previous one because here we have unequivocally angiosperm remains. I include here these two, Braseniopsis and Cycites, observed and described by Carlos Teixeira. In this flora, I was there. This flora is Buarcos Tavarede. Of course, these sites disappear because of the urban development, and many of the sites are covered by the motorways. And also the angiosperms, there are also remains assigned to ferns and conifers with clearly xeromorphic factors. Regarding to these swans, to Braseniopsis and Cycites, according to Teixeira, they belong to the lineage that diverged near the base of the group or near the base of angiosperms group, especially Nymphaeales and Ranunculaales. According to Teixeira, these leaves were very thin to float on the water, and in Teixeira's opinion, these two plants were aquatic plants. Braseniopsis is related to Nymphaeales and Cycites related to Ranunculaales. Here we have two more specimens from the Buarcos Tavarede flora. There are leaves. These leaves was interpreted by Saporta as Sassafras, Protophyllum, but Teixeira does not agree and put it in another genus, in the genus Aralia. However, in my opinion, this is very confused and this material requires a revision. There is another important reference in this flora, but I did not see the specimen because many fossils disappear from the collections. But Teixeira, in his book from 1948, he makes reference to the occurrence of magnolia leaves. I saw the photo in the plate, but it's not very clear for me. It's a big leaf, but the photo for me is not very evident. I don't know if I saw if I refer or not. This flora, the circle flora, belongs to the Apsian Albian age, but according to Teixeira, this one is a little younger. Is from Albian. Well, after these studies based on macrofossils or leaf fossils, the studies about paleobotany here in Portugal was more or less stopped during several years. But in the 60s, several researchers started with palynological studies. And this was important because palynological studies have added important information on broader patterns of vegetation change through the Portuguese Cretaceous. The palynological studies were focused mainly in the late Cretaceous, but there are also a few studies on the early Cretaceous too. Regarding topological studies, we have a little problem because when we are using the light microscope, there is a challenge to find or determining the systematic affinities of all isolated or dispersed pollen and spore. And the taxonomic resolution sometimes is very difficult and is not possible below to the order or family level. After the palynological studies in the 19th, this lady, Else Marie Fries, opened a new window in to the new world of the angiosperms or early angiosperms evolutions. Her studies were developed mainly here in Portugal and focus on the reproductive structures of angiosperms, flowers, fruits, seeds, and dispersed stamens. And these fossils comprise very informative factors about the first angiosperms of the Cretaceous period. Elsa Fries was my PhD supervisor. I was her student. And when we decide to start with the study of the mesofossil floras from the early Cretaceous of Portugal, we decide to not focus only on the angiosperm mesofossils, but in each flora we collect everything and we study everything. Why? Because we have been used the floral composition or plant community composition, as you want, in order to get information about the environmental conditions in which the first angiosperm developed. So the Portuguese mesofossil floras are known from the Berriasian to the Campanian Maastrichtian periods. The earliest and well-described angiosperms are from the late Barremian, early Apsian from Torres Vedras region, and the excellent preservation of the mesofossils has allowed to make detailed comparisons with the extant plants and enable the taxonomical studies and has provided new insights into the reproductive biology of Cretaceous angiosperms, such as some kinds of different kinds of pollination types. There is an important point regarding to the mesofossil floras because they are spread in the Lusitanian Basin, since the Torres Vedras until the north of Aveiro is a very large, is a very extended area. But a detailed knowledge of stratigraphy or lithostratigraphy does not allow to date with good resolution all continental facies. We have collect all only continental deposits, but in some floras the age is not very easy. And especially because we cannot, it's not easy to establish correlations with the well-dated marine units next to the sea. Besides, in these mesofossil floras, we haven't got biostratigraphic markers, so sometimes we need to use larger intervals, and I confess that this bothers me and I do not like to do it. Here I have a summary of the most important sites. There are others. Fossil sites in the Lusitanian Basin for leaf floras, mesofossil floras, and callinifloras. Here in Bordeaux, or red color, I include the early Cretaceous mesofossil floras, Sais de Barresian, Valpeinho, Autorivian Valcortiço, Barremian Casal do Borracho in Torres Vedras, and in the late Apsian early Albian, in this interval, we have several floras, mainly in Juncal area, Val Aqua, Catifica, and Buarcos. And finally, in Cercal, we have Apsian Albion, but in Cercal, we only have the leaf floras I have referred before. I was there. I have tried to find the sites documented by Saporta and by Carlos Teixeira, but it was impossible to collect material. Then in green color, we have the late Cretaceous floras, some from Cenomanian of Vila Flor, Vila Verde, and Tentugal, very close to Coimbra. These sites does not exist anymore. I collected material from one Cenomanian flora next to Coimbra, but it's not from this site. It's from Condeixa, and all the samples was completely barren. Then there are also the Turonian mesofossil floras from Tentugal, Carrajão, and Requeixo, and the late Coniacian from Ceador. And from this flora, I have very, very, very nice material. I found recently the first seeds assigned to the Andresianthus fruits with very beautiful normapoles in situ. And then we have the late Campanian Maastrichtian floras from Mira, Preza, and Esgueira. Elsa Marie Fries was here in these three sites. I was here, but only Esgueira is available to collect material at this moment. So here we have the Lusitanian Basin in more detail. Here you can see these green shots. Here we have Catifica and Torres Vedras. Then we have in the Leiria region the Juncal, Valacqua, and Famalicão floras. I have worked mainly here in Juncal and Valacqua. Then here, next to Figueira da Foz, we have Taverede, Vila Verde, and also Buarcos flora. More in the north of the Lusitanian Basin, we have the Myra, Preza, and Isgueira. And here we have also the new flora from the Coniacian in Ceador. Well, In this figure or in this diagram, I include the flora, the mesofossil floras studied and documented until the moment. There are more, but I selected this 1, 2, 3, 4, 5. And I include also Theodoro and Isgueira because I was there, but I only the work is in progress now. As you can see, Valpainho is in the base of the early Cretaceous. The age is Berriasian. Then we have here Val Cortiço from the Autorivian. Here Casal do Borracho. The age is assigned to late Barremian to early Apsian. Here we have Catifica. I put Catifica here in Apsian and not in the interval, and later I will explain why, because in my opinion, we cannot include this flora in the same pack as Juncal and Valdacqua, because Juncal and Valdacqua, we consider the age between the range late Apsian, early Albian. I'm sorry, there is a small mistake in this figure, but I could not correct it because I adapt this from Ray et al. 2006. I put Juncal here in Albion because Figueira de Foz formation is here too, but the interval should be a little bigger and start in the late Apsian until the early Albion. Then we have the Theodoro, Coniacian, and Isgueira, Campanian Maastrichtian. Well, is the here we have a nice picture from the first clay pit complex in Juncal. This is Valpainho clay pit complex. The age is Berriasian. Is the oldest mesofossil flora we have. And we collect material here in this mudstone layer, as you can see here. And this is, we did not find any remains of angiosperms, but we obtained very good results in this flora. Besides, there is another important question because this flora, Valpainho mesofossil flora, is the only one in Juncal area that are outside of the Figueira de Foz formation. All floras in Juncal area are included in here, in Famalicão member from Figueira de Foz formation, and the age is early Albian to late Apsian. However, we have an exception here because we have some kind of crockets incline and also an unconformity, and we have here the upper part of Bombarral formation, and the age of this flora is probably Berriasian, and the Valpainho samples come from here. Well, this flora is completely dominated by the gymnosperm remains. There are several conifer seeds and twigs. Some of them is difficult to say what they are because they are very distorted. But in this flora, we found very interesting specimens like these ones I have here in these plates. Here we have specimens assigned to the Hermannspermon genus, previously described by Else Marie in Bornholm, Iceland, in Denmark. She described another species very similar to this one as Erdmanispermum balticum, sorry. And here we have another one. And we described this as a new genus, and the species assigned to Raunsgardispermo lusitanicum. In my opinion, and I will talk about this later too, these seeds are more important than we can sort. Many people does not agree to talk about this as belong to the BEG group, Benetitales, Hermanitales, and Genitales group, but these seeds are very important. Also, they share the same organization plan. There is an outer seed envelope, there is an integument and a nucellus, and typically they show a pointed micropylar region, as you can see here. Here in Hermanspermon, as you can see, we have a three-valved seed, but here we have a bivalved, okay? But there are four-valved too. The diversity is higher. Regarding to the because we, as I've told you before, we are study everything, and we combine the palynological studies with the mesofossil flora study. This Palinoflora is very interesting too. Is completely dominated by fern spores and gymnosperm pollen. Among the spores there are the Cyatidites, Crucisporites, cicatricospores, Pilosysporites papillosos, and among the pollen grains, there are many. The flora is dominated by Classopolis pollen grains. There are also bisaccate pollens and, of course, Eucomidites pollens. Well, the palynological assemblage from Volpino is dominated by pteridophytes and gymnosperms. The mesofossil flora is dominated by conifer seeds and seeds assigned to BG group. The associations indicate, in our opinion, the presence of a conifer forest dominated by Caryopodiaceae with the ground cover and the understory vegetation composed by ferns, as well as members of the BG group plants, and no angiosperm remains were recognized in this flora. This flora is very, very, very similar to this one, but they share similarities with other contemporaneous floras from the North Europe, but mainly with this one from Bornholm, Iceland, in Denmark. Else Marie described this the flora from Bornholm and the the similarities are high. Well, this is another mesofossil flora from the Eterevian, is younger, little younger, and here we are in Valcortiso Clay-Pit complex in Amiel, close to Torres Vedras. This flora is it's very nice. This clay pit does not exist anymore because it's infilled now. And this flora is very interesting, in my opinion, because it's dominated by gymnosperms, non-angiosperms, and there are two genus very common among the Valcortiso flora, Phrenelopsis and Pseudophrенelopsis. We amended the original diagnosis of the genus from using the material from Phrenelopsis teixeirae recovered in 2010 from Valcortiso flora, and more recently we described this beautiful Pseudophrenelopsis as Pseudophrenelopsis denisi. And more recently, we found this very, very interesting male cone with imbricate microsporophiles arranged helically, and in situ, we observed several classopolis, pollen, very beautiful, that in my opinion belongs to, could be assigned to the species Classopolis martinoti. Besides, we can relate this cone, in my opinion, with this species here, Phrenelopsis teixeirae, because for two reasons. Um, both the phrenelopsids are common in the flora, but Phrenelopsis teixeirae dominate clearly, is very, very, very common. And also the stomata arrangement in Phrenelopsis teixeirae is very similar uh, to which what we observed in this male cone. So probably we have here a link. Phrenelopsis teixeirae probably produce this cone, Classostrobos amielensis, and Classostrobos amielensis produces pollen grains assigned to Classopolis martinotti, and I think this is not speculation. In this work, we count with the collaboration of my friend Tekleva, is in the audience, I think. I have studied the Palinoflora from Valcortiso in detail. This Palinoflora is interesting too and diverse. We have like Triradites, we have Patella sporites. There are diversity of cicatricial spores, Concavissimi sporites, Cyatidites, Australis, Pilosis sporites, Ceratosporites masculus. Among the pollens, we have several Classopolis are the dominant group of conifers, Spheripolonites psillatos, Araucariusites australis, Calielasporites dampieri, Calielasporites trilobatus, Balmeopsis, and several bisaccate pollens. This bisaccates is difficult to identify because sometimes the dimensions of the bodies comparing with the suckles is not very clear. Well, the Valcortiso mesofossil flora is dominated by conifers, assigned to the species Phrenelopsis teixeirae and Pseudo-Phrenelopsis denisi. The Palinoflora is dominated by fern spores and gymnosperms, pollen. However, bryophytes and lycophytes are present in the pollen spore assemblage, but subordinate. Similar to what we found in Valcortiso, in Valpeino, here in Valcortiso, no angiosperm remains were recognized too. The composition of this flora points to the presence of an open vegetation, clear consisting or composed by a mixed conifer forest with an understory ground vegetation dominated by ferns. So riverine environment with surrounding vegetation of open woodland and ground cover, primarily of ferns, is strongly indicate for the region. However, in my opinion, the wet periods are seasonal. Here we have another important site, another important flora is the well-known mesofossil flora from Torres Vedras, and this site is Casal do Borracho. The age of this flora is late Barremian to early Apsian. Elsa Marie was here in the 90s. I was later, and the site is completely different. I could collect some samples here because I could catch part of the mudstone layer, but not all, because behind of these people here there are many houses, and this site is completely different now. Besides, my mesofossils recovered here in Torres Vedras are not so nice as the ones collected by Elsa Marie. I could find some or identify some conifer seeds, some BGs, and several angiosperms remains, but not so not very well preserved. So a good point here is about the oldest angiosperm remains in Portugal. In my opinion, it's important to say this because several times I have read in several papers that the oldest angiosperm remains in Portugal correspond to dispersed pollen in the sediment. And when they say that, they consider pollen grains from or these pollen grains assigned to Clavate pollinites ugesi from the Valanginian of Porto da Calada in Ericeira. This site is located next to the sea, and in my opinion, is very hard to believe in this, and I will explain why. I was several times in this site in the Porto da Calada. I collect several samples in the same fossil layer from the Valanginian age. I have prepared several residues. I prepared many alien slides. I observed all the slides in detail. I also put some drops in the stubs for observed in SEM, and I never found any grain assigned to Clavati pollinids or other angiosperm. Besides this Clavati pollinites ugesi, it's only one grain and appear in the PhD thesis of my colleague Trincão, who does not work in paleobotany anymore. So, in my opinion, of course, this is my opinion, this was a case of contamination. Well, after this, angiosperm remains were only found again from the upper Barremian to lower Aptian of Torres Vedras. And from here we have this log. This log is in my paper from 2080, where I described this flora, and the samples were collected exactly in the same site where Elsa Marie collected the samples for the mesofossil flora studies. But as I have told you, my mesofossils were not very nicely preserved. And the age of this site is clearly established by Hay, Jacques Hay, from his late Barremian, early Aptian, and belongs to the Almargen formation. Well, here we have some, of course, I cannot show you everything about the mesofossil floras. Here we have some bisexual trimerous flowers with retimonocolpites type pollen on the stamens. These come from Torres Vedras. The flowers are not complete. Here, another interesting specimens. As you can see, there is a fragment stamen or an anther. And here we have Clavati retimonocolpites pollen grains in situ in anthers, and coprolites from Torres Vedra also. Elsa Marie studied several pollens observed in situ in coprolites. Here, another interesting specimen, Apomatoxia, with the characteristic spines. Some fruits with small spines and other with spineless and related with the Xanthipperales. The pollen, here we have an anther. The pollen grains observed in this Apomatoxia are assigned to Tucanopolis, to transitory polis. Here we have Tucanopolis transitory polis, here a monocolpate pollen, and here a finely striate and monocolpate pollen grain. This material is from Elsemari. Here, more beautiful pollen grains. All these pollens were observed in situ in coprolites. Here we have polyporate grains. Here we have polyplicate pollens assigned to Mayoa portugalica. I found Mayoa in my samples from the palynological studies. Here we have a tectate pollen similar to the extent Acroatia. Here we have in a coprolite several types of pollen grains. We have Clavatipollinites, Retimonocolpites type pollen, Tucanopolis transitoripolis pollen, and Pennipolis pollen grains with characteristic spinose tectum. And here we have a monocolpate pollen. But most important, Elsemari only found these two grains of tricolpate with of three germinative apertures. So from Torres Vedras we only have these two pollen or these two tricolpate pollen grains. That could be an evidence for the presence of eudicots. Here are the results of my Polinoflora studies. This Polinoflora, which was studied in detail, and it is very interesting. Here we have again Tirarites, here Taurocosporites, Clucisporites, Patellasporites, Hietidites. Here a spore from an fresh algae. Water is Ovoidites prigi. Here we have cicatrical spores, and among the pollen grains we have Arboricites, Callilasporites dempieri, Bissacates, pollens, Classopolis, and I found in the slides only Clavatipollinites pollen. But I put the residue in the stubs and observed them in the SEM, and I could, of course, I observed the same spores, but among the pollens there are also Eucomidites and Clavatipollinites, and here Retimonocolpites, again with this beautiful grain with the spinose tectum. Well, unequivocal unisexual flowers, very similar to those of extant Ediosman, are common in Torres Vedras flora. I put here unisexual flora. I should put fruits because really we have complete fruits, not complete flowers. Angiosperm pollen grains recognized from Torres Vedras mesofossil flora is monoaperturate and probably reflects the presence of plants closely related to Cretaceous, as well as monocots, and perhaps various eumagnolid angiosperms such as piperoles. As I've told you before, we only have two different tricolpate pollen types, provide the only evidence of the presence of eudicots in Torres Vedras assemblages. Besides, in the coprolites were found several Pasteropolis, Clavatipollinites, and Pennipolis. Regarding to Pennipolis, Elsemari relate Pennipolis with Alismatalis, but in my opinion, I think these Pennipolis are most related to an extinct lineage, according to Doyle and Autumn analysis, would be most related to an extinct lineage related to Cretaceous, since they lost the columella. The several taxa of Cretaceous from Torres Vedras, including plants similar to Ediosma, are also represented in the dispersed pollen, notably represented by Asteropolis. The occurrence of several coprolites rich in pollen, including Clavatipollinites, shows that some plants in this range or in this period were visited by insects, and the overall and the Palinoflora results suggests a locally wet environment in a tropical subtropical climate, with much of the gymnosperms component indicating more arid condition, but not very far away from the time of the deposition. Here we have my favorite site and my favorite flora is Catifica, and I put here the Catifica outcrop section with the age Apsian Albion. But here we have a story, and I'm not completely agree with Elsa Marie. I understand what her arguments. Elsa Marie would like to narrow the interval late Apsian, early Albian, but I do not agree with her position regarding to the age, because according to her interpretation, this flora could be younger than Apsian, but I do not agree, and I will explain why. Because if you look with attention at this picture of the outcrop section in Catifica, we have here an unconformity, and then we can divide this in two parts. This is my interpretation according to, and is in accordance to Jacques Rey ideas. Jacques Rey is not between us anymore. He passed away a few years ago, unfortunately, but he was several times with me in Catifica site. So we have here an unconformity, and we have a lower unity here with these fine granite sandstones with white color, with intercalated with these mudstone layers very rich in plant remains. And in the upper unit, we have this yellow and coarse sandstones. So according to Jacques Rey, this lower unit could correspond to the lower member of the Almargem formation. And if is it correct, this age is Apsian and not Apsian Albion. And all the samples which we have from Catifica were collected here in these levels or here by Elsa Marie Fries. Well, Catifica is a very rich flora. Here we have some gymnosperms. I will not talk very much about the gymnosperms because we are preparing with Elsa Marie monography. This is a work in progress. I don't know when the monography will be ready, but I hope so. Here we have some phrenelopsids from Catifica. I feel that are Phrenelopsis turolensis. Here, Pseudophrenelopsis parsermosa. We have also Watsonioclados virginiensis and Pagophyllum. This specimen is very interesting. Was collected in Catifica and solve a problem for palynologists, and I will explain why. The cone is very beautiful. Is an coriaceous cone. We have described it as a new genus and species, Callialastrobososai, In this cone, we found many pollens in situ and pollens from two different genus. We found Paracariacetes australis pollens, but we found also the Callialasporites with the characteristic monosac around the pollen grain, Callialasporites or Planospis dampieri. And we solved this problem. Why? Because for some palynologists, during many years, Callialasporites were assigned to the Porto and to another ones for assigning to Coriaceous. But with this finding, we would conclude that this cone produced these two kinds of pollen grains, Arocaricetes and Callialasporites. So Callialasporites is definitely an Arocariaceous pollen grains. Well, Catifica is very rich in these seeds related to BG group. I feel that we found about 12, 13 species. I'm not sure because the last one we described it was Botanispermo hirsutum. I have described it with Else Marie and Kai Ronsgaard Pedersen in 2019. But in this flora, these seeds are very common. Here I have a very beautiful specimen from Buarcospermum tetragonium. The other species I have shown you from Juncal were 3 and 2 valved, but this is 4 valved. And as I've told you, these seeds share the same pattern of organization: an outer envelope, an integument, and an ocellus, and also a pointed micropylar area. In my opinion, these seeds are very important, or most important than we can sort in the first view, because for many researchers the Gnetales and the Benetitales are related to the gymnosperms, and I do not know if this could be or not a link between the gymnosperms and angiosperms. But we have got a problem here. In these seeds, it's very difficult to find pollen in situ in the micropyle. I can tell you that I have mounted about 40 specimens of Buarcospermum tetragonium, and I never could find any pollen grains. And regarding to the other species, as for example Lobospermum, the same happens. I cannot explain why. But we haven't got male structures in this flora, and also we haven't got the seeds with in situ pollens. Well, regarding to the angiosperms, here we have a very beautiful specimen assigned to the Chondritia. Chondritia is related to Cretaceous. Here we can see in the fruit the characteristic resiniferous bodies. Here a fruit with 2 seeds inside. Here we have a seed, and here pollen grain assigned to Piercipolis. Elsemarie include this in the genus Piercipolis because in the type species from Retimonocolpites, the colpos appear in the distal phase, but also in the proximal phase. So the colpos is around of the pollen and divide the pollen grains into alphas. But honestly, I do not believe in this anymore because I do not know if there is some kind of polyploidia in the meiosis or not. Because if we compare the reticulum, they are very similar. For me, this is everything assigned to retimonocolpites type pollen. Here we have another seed of Chondrite, but different species. Is Chondrite fovulata, is very common with the with this pitted outer surface. And here we have a beautiful staminate structure assigned to Proensystemon portugalicus. This staminate structure is composed by several tetrasporangiate stamens and was studied in detail with us, including Maria Tecleva. There is a paper in which we have made a detailed analysis of these trichotomocolpate pollen grains before we described the specimens as Proensystemon portugalicus, and our results regarding to this. I think the three-armed sulcus is not very clear here, but I confirm that is true. And Maria is here in the audience, and she can confirm what I'm saying. And these three-armed sulcus could be an intermediate stage between the old or the ancestral simple corpus of Quarantaceae and the 4 to 6 armed sulcus observed in the Asteropolis, and could be a good key to relate this to the extant Neotropical Ediosmon. Here we have a fragmented stamen with 4-armed or tetracotomocolpate pollen. Here, Clavatipollenites pollen grains. There are a great diversity of these Clavatipollenites and also retimonocolpites in this flora. Here we have very beautiful seed fruits assigned to the Cerealis genus. Here two different species, Cerealis communis and Cerealis crassitesta. When we start the studies and when these seeds appear, we saw that we only have 2 species, but there are more. Because in Juncal, for example, and Valacqua, there are seeds clearly different to these ones, and of fruits very different. Not here we can see 1, 2, 3 seeds inside, but I have fruits with 4, with 5 and 6 seeds inside. These fruits are related to Magnoliades. Here we have a very common flower from Catifica. We have here Saportanthus genus. I'm sorry, I not exclude this flower because this and this belong to the same genus, but the species are different. This is Saportanthus parvus and this is Saportanthus dolichardinum. This flower is not from Catifica, but I include it here because these pollen grains here was in situ here, okay? And these pollen grains are similar to the ones we can observe it here in this flower. The pollens show a very thin wall. They are finely microstriate, and in light microscopy, they not seem to be pollen grains, but fungi, because these pollens are, I cannot explain. I never found these pollen grains in the dispersed pollinophora. I only could see this in situ in the flowers. This flower was related to or included in the Laurales order by Elsemarie, but we used the Doyle and Andrews dataset, and Maria Tecleva collaborate with us in this work too, and we conclude that this flower could represent an extinct lineage of the early angiosperm flowers, which is not the same to say that this flower is an Laurelian representative. Here in this figure, we have, in this plate, we have trichoepate pollens. In Catifica, we have several types. Here we have another beautiful flower. Paigia pentoporata is a flower with the pentamerous organization, 1, 2, 3, 4, 5 in X-ray. And in situ, we found these pollen grains, pentoporate pollen. Here we have another type of trichoepates, and of course, in Catifica, the diversity of trichoepate pollens is higher when compared with Torres Vedras. So the eurycodes were very well established here. Well, the Catifica mesofossil flora is diverse, and in addition to angiosperms, in this flora we can find also many non-angiosperm fossils as lithiacs of bryophytes, sporangia, fragments of ferns, twigs, cones and cascades of conifers, and seeds assigned to chlamydospermous seeds or seeds related to BG. Angiosperms dominate in number, but we must be careful with this because we don't know yet how many specimens exactly and how many species of non-angiosperms we have in Catifica, because as I've told you, we are preparing a big monography about the non-angiosperms from Catifica, and we not until the end at this moment we did not know the exact number, but I believe that angiosperms could dominate, but the number could be very, very, very thin, perhaps 50-50 or 60% of angiosperms, 40% of non-angiosperms. Angiosperm diversity is mainly observed at the level of non-anticots, including anagrade, angiosperms, chloranthaceae, and magnolids. And of course, the chloranthaceae are the most abundant representative. Remains of chloranthoids, only for your idea, are especially common in both number of specimens and also in number of species. We have about 40%, the chloranthoids represent about 40% of all specimens of angiosperm specimens recovered in Catifica, and more than 25% of species recognized in Catifica flora. The Catifica pollinoflora is very rich. I have published a paper last year with the preliminary results. Is diverse and dominated by fern spores, assigned to Anemoniaceae, Cetacea, Dixoniaceae, Liquidiaceae, and Osmundaceae, reflecting clearly the existence of the presence of swamp pteridophyte vegetations. And we have also the occurrence of bryophytes and lycophytes represented by Taurocorsporites, and Neuristrischia. This proves or this supports the presence of wet, humid conditions in this flora associated with small lakes and alluvial plain. The abundance and the occurrence of pollen grains of the genus Eurycoecites mainly reflects the presence also of the conifer-dominated vegetations that cover relatively dry areas in the inner islands. Angiosperm pollens are less frequent in the dispersed pollinoflora, but they are very well represented in this pollinoflora. And I can tell you that of course, the genus Calavati pollinitis is very common, but I found several trichopates pollen grains types. Then here we have another site from Juncal area. This floor here you can see an overall view of Chicalhão clay pit complex. The age is late Epsian, early Albian. This is in the base of Famalicão member from Figueira da Foz formation. We study the polinoflora and the mesofossil flora of Chicalhão. The polinoflora is clearly dominated by fern spores and gymnosperm pollens, but angiosperms are represented too. Here we have Ceatidites, cicatricols, here Costatoperforasporites. Among the gymnosperm pollen grains, we have different types of Classopolis. We have also Bisacates. Here we have two different, here Clavatipollinites, and here two different types of Retimonocolpites pollen grains. And also we have here the genus Rosia represented in Juncal flora, in this first flora, and Senectotetradites. Regarding to flowers, we have found 4 types of flowers here in this flora, in Chicalhão flora. These 2 ones are not very well informative. Here we have the dolish, we have supportant, sorry, I'm a little tired now. We have Supportanthus dolichos temon species with these typical pollen grains, and here a very beautiful flower described for the first time in Europe and assigned to the Zebaliaceae ranunculates. And I will explain in the next slide what we found exactly with tricolpate pollen grains. Here we have fruits of Chondritia, here Chondritiopsis with retimonocolpite stubborn grains in the region, in the stigmatic area. In this mesofossil flora, we found these typical seeds related to the Nymphaeales, and also these ones. And here we have the flower I showed you before. We describe it separately before, after, sorry, as Caianthus lusitanicus. In this flower, we can clearly see 3 free carpels, 6 stamens, with robust anthers, and the sweet characters observed in these flowers, including the pollen grains, tricolpate with the polygonal ornamentation. Based on these characters, we could include it in the Lardizabalaceae family and in Ranunculates order. And these flowers were described for the first time in Europe in Juncal. This flower is related to the extant Sinophrynchestia chinensis from China. The polinoflora from Chicalhão is dominated by conifer pollen and fern spores. Angiosperm pollen is also present but subordinate amongst the polynomorphs. In contrast, and similar to what happens in our floras, angiosperms completely dominate the mesofossil flora, but not dominate in the polinoflora. 4 taxa of angiosperm flowers were identified, and 1 flower is assigned to the Lardizabalaceae, closely related to Sinophrynchestia endemic to China. The plant community indicates seasonally dry climate and an open vegetation consisting of a mixed conifer forest with an understory and a ground cover of ferns and angiosperms with a patchy occurrence of lycophytes and also plants related to the DG group. Angiosperms probably inhabit moister area bordering rivers and lakes in this region. Here we have another flora from Juncal area. This is Nossa Senhora da Luz flora, and this flora is also included in the same range of age, in the same interval, late Epsian to early Albian. The mesofossil flora is the polinoflora is very interesting. It's dominated by ferns and spores and gymnosperm pollen. Among the spores, we have cicatrical spores, we have Ceatidites, Glycosporites, Patellas, Todisporites. Among the pollens, we have different types of Classopolis too, Bisaccate pollens, Araucaricetes, Calliolasporites, and Acomedates. Well, in the angiosperms, we have these beautiful brains, Stellatopolis bergoni, with this crotonoid ornamentation. This flora, Nossa Senhora da Luz flora, is very, very, very rich in this kind of pollen grains. And I do not know why. I have collected several samples, but I never find the flower or the plant mother of these pollen grains. And honestly, I'd like to know which kind of plant could produce these pollen grains. This kind of ornamentation could also indicate some kind of specialized pollinators where that were active during the early Cretaceous. Well, in Nossa Senhora da Luz and among the angiosperms, we have also this Senectotetradites, an obligate tetrad, and Afropolis zonatus with this zona sulcate pollen grains. So we have here two Gondwanan elements, and very common. These two kind of pollens are associated to xerophytic floras and to hot and dry climates. Here in this side we have Clavatipollinites, monocolpate pollens, and 4 different types of Retimonocolpites pollen. Perhaps you cannot see very well here, but I can assure that the ornamentation of the reticulum is not exactly the same in these 4 kinds of pollen grades. Among the mesofossils, we have lycophytes here. We have the selaginellatious megaspores are very common. Brachyphyllum, conifer seeds. Here we have Anacostia seed. Here Anacostia fruit. Again, Chondritia. Here we have Chondritiopsis, another flower of Saportanthus dolichostemon, and these typical seeds of that could be related to the Nymphaeades. Here, other cerealis. This is a little different from the one from Catifica, and there are also two another types. The pollinological assemblage of Nossa Senhora da Luz is diverse, including 588 spores and pollen grains assigned to 30 genera and 48 species. This pollinoflora is dominated by fern spores and conifer pollen. Angiosperm pollen is present, but subordinate among the pollinoflora. The mesofossil flora is less diverse, including 175 specimens assigned to 17 species and dominated by angiosperm fruits and seeds. The mesoflora also contains conifer seeds and twigs as well, and also several selaginellatious fossils, namely megaspores. The fossil assemblage indicates a warm and seasonally dry climate for the Nossa Senhora da Luz flora. Well, in this figure, we can see that from Torres Vedras until Juncal is a clear evolution in the angiosperms floras. In Casal do Borracho, of course, we have eudicots represented by two pollen grains, but when we go from the Casal do Borracho until Juncal, we feel that the diversity of angiosperms growing from year to year. There is here we have another point that bother me is the interval, the bigger interval for all the floras, because of course I did not show you everything, but in Juncal area we have several floras. There is Carreguera flora, for example. I did not show you. There is another one which we are studying now, Mayorga, and all floras are in the same interval. But in my feeling, I feel that even in Juncal, if we check careful the floras position in the base of Famalicão members from the Figueira da Foz formation and the other ones, for example, the Nossa Senhora de Luz flora, there is a difference. We are trying to narrow this interval. Elsa Marie suggests to use sediments, but I don't know if it will be possible or not. Well, general conclusions about this. Plant fossils from Portugal and Spain unequivocally document the presence of angiosperms in Portugal and in the Iberian Peninsula around the late Barremian to the early Eocene. The systematic studies and comparisons with the extant plants suggest the earliest forms were herbaceous vegetations or small shrubs, constituting the ground covers in different floras. The aquatic habit was established early throughout Iberia. In Portugal, the earliest forms are represented or are related to the Nymphaeales, and in Spain are also represented by Ranunculus ferreri. The specimen's diversity in the mesofossil flora is higher when compared with the leaf floras and with the dispersed polymorphloras, which can be explained by the low fossilization or the low preservation potential of the herbaceous plant leaves that are very fragile. The number of angiosperm pollen grains among the dispersed pollinofloras is lower than expected, and at least when I start with this, I expect to find more, and this could be explained, or this could be related to the insect pollinated plants. Early angiosperms included taxa to Cretaceous. Monocots were also abundant in the lower Cretaceous, confirmed in Portugal by inflorescence, flowers, and dispersed stamens with in situ pollen. The first eudicots are reported from late Barremian to early Albian of Torres Vedras, and afterwards they diversified in the late Eocene to early Albian. And finally, the mesofossil floras from the lower Cretaceous of Potomac group and others and other regions in Europe share similarities to the ones of Lusitanian basin. But there are, regarding to the early Cretaceous mesofossil floras, there are strongly similarities to the ones studied here from the lower Cretaceous of Potomac group in the other side of the Atlantic Ocean. Of course, in the late Cretaceous, because of the separation of the continents, now they are in different geographical positions, and the floras, of course, evolved in different ways. So many thanks for your attentions, and sorry, I think I was very extensive in the time.
Thank you, Mario, for your beautiful talk, full of information, full of images. I have enjoyed it greatly. And now we can ask questions. You may write your questions in the chat, or you may raise your hand like this, so with an artificial hand, and I'll give you the word. Well, so far I see only greeting in the chat from Stephen Manchester. Well done, thank you very much. And from Maria Tekleva, very interesting talk, thanks Mario. And so far I see, I see a hand, I see applause by Julia Gravendick. Well, okay, I'll ask you, I have in fact several questions. Mikhail Stanislavovich, да?
Yes.
Okay, Mikhail asking.
My question is not about angiosperms, sorry. About bryophytes, it was about Tauricus paritis.
Yes.
You refer to bryophytes. Why? My question is because it's sometimes somehow difficult to attribute maybe Tauricus paritis to one of
Yes, I discussed this with I think you are referred to Taurocos porites from Torres Vedras. I discussed this with David Button, and we agreed they belong to bryophytes, because in the literature these spores are assigned to the bryophytes, and there are similar spores from the extant plants very close to the Taurocosporites.
Thanks. Is it somehow related to finding of Benjamin Bonfleur with his bryophytes?
Yes, I know.
Probably,
He studied with us.
Is it the same place?
He studied with us some material from Catifica too. Yes, but not spores, only mesofossils.
Thank you very much.
Thank you. Next question. Well, okay, I can ask mine. You mentioned coprolites and pollen grains inside coprolites.
Yes.
Was it a mixture of pollen types or one and the same pollen type?
Sorry, Natalia, I do not understand very well. What do you want? I never put the coprolites in the microscope to see what there are in situ, but Elsa Marie did it, and she found very pollen grains. As I show you in Catifica, there are several pollen grains in the coprolites. But please repeat your question, because I did not understand the second part.
My question was, are there several types of pollen grains in the same coprolites? Or there was only one type of pollen?
No, several types, Natalia.
Several types.
Several. Clavate pollinitis, for example, in Torres Vedras, we found in the same in a clump of coprolites. She found, for example, 4 types: Clavate pollinitis, Retimonocolpites, Tucanopolis transitoripolis, and also Penipolis.
Thank you. I was interested if this pollinator was restricted to one type of plant or visited several ones.
Yeah, and because of this, we could relate the presence of the pollen grains in the coprolites with insect pollinated plants.
Thank you. I see Tatiana Alexeyeva wants to ask something.
Yes, thank you very much. So my question will be about your excellent images, electron microscopy images.
Yes, thank you.
May you say something about the procedure for preparation to get such beautiful pictures?
Where I prepare?
How?
In Sweden, Stockholm.
No. What was the way? What kind of treatments have you done before scanning?
Oh, what kind of, okay. The mesofossils need to be cleaned with HF, and then we mount the specimens in the aluminum stubs, and then they are coated with gold.
So you have not treated with acids or strong chemicals, just water, or?
No. I saw as I saw the specimens when I sort the specimens in the stereo microscope, and then the best specimens are all clean with hydrofluoric HF acid.
I see.
Okay. Yes.
Okay. Thank you very much.
Not at all. Not at all.
Thank you. Julia Gravendick, please.
Thank you, Mario, for
Hi,
your
Julia.
beautiful, extensive overview and so many nice images. You often refer to things as retimonocarpitis types and clavatimonocarpitis. Since we're also working on this, I find it always baffling how much diversity there is in these genera, where you lump a lot of different taxa. How do you approach this? How many differentiations do you try to do? Do you stick with a type version, or do you actually, for yourself, even if not
No, on the basis of the
more?
no, Julie. On the we conclude that they are different on the basis of the ornamentation of the reticulum.
So have you tried to take some more detailed pollen out of the mesofossils to make some more detailed preparations, for example?
No, and because of this, I told you regarding to the PRC polis, I don't believe in that story anymore, because for me, PRC polis and the retimonocolpites are the same. Because the story about the corpus don't convince me. Because I don't know if during the meiosis process there are some kind of polyploidy or another kind of mutation. Because when I saw the pollens, when I compared at least the reticulum structure, they are very similar. The only difference is the position of the corpus, because in retimonocolpitis, according to what you can read in the type species description, the corpus is around of the pollen and divide the pollen grain into alphas. And for example, the pollen grains in the Candritia, you see the corpus in the distal phase, but the reticulum is similar. And for me, this story, I don't know, for me is all the same. In my opinion, I think these two genus could be the same, retimonocolpitis and no more.
Thank you.
Thank you.
Not at all, Julia.
I see manual hand. Xinkaiwu, please.
Hello. Thank you for your nice talk. I am just curious about your conclusions, because when we do the Cenozoic, when we do the paleoclimate reconstructions, we will compare with the living plants, and based on the environment, we will say the paleo environment is climate, is humid or dry. But when I listen to your talks, I see several conclusions. It said it's seasonally dry climate, but several fossils is a new journals, and how to get to the conclusion is the seasonally dry climate, and how to do the paleo environment reconstructions in the for the Cretaceous plants.
Well, we are using, if I understand, you do not understand very well why there are seasonally periods. Is this?
Yeah. Why is it seasonally dry climate and which characters can indicate it?
We are using the as proxies the specimens, the plants, but we are also, I have no time to show you everything, but we are also use some sediments, okay, some minerals. Okay, and some of them are in accordance with our interpretation. Besides, as you know, in the final part of the Jurassic, the climate was very hot. But during the Cretaceous, there was some kind of fluctuations. You have some periods more hot and the other ones less hot. But these seasonal periods that we conclude there are in the floras regarding to the wet periods is related to the small vegetation, mainly the ferns, for example. Okay, but we have also some minerals that point to that. Okay.
Okay, thank you.
Thank you. I see several comments in the chat. Excellent talk from John Marshall, Eduarda Martinet, Evgenia, I suppose, Bugdaeva, Elena Burkanova, Emmanuel Rui, and I also see a question from Bruce Cornet. Excellent talk, Mario. How do you know they are coprolites? Insect coprolites?
It's very easy to identify the coprolites among when we are in the stereo microscope. It's very easy. They have a characteristic shape, ovoid shape, and there is no doubt. Of course, when I saw it for the first time, I did not know what is it. But during the time, I was understand there was coprolites. Sometimes they appear isolated, sometimes they appear in clumps, but they are very characteristic.
Thank you. Well, do we have more questions? Well, if not, I'll ask one more. You mentioned Normopolis pollen in situ.
Yes, I did not show you because the talk was very, very extensive. Yes, I found very recently, I found very nice fruits from the Coniacian flora. These fruits were described for the first time from Else Marie, but not from the Coniacian, is from Campanian Maastrichtian. And the fruits in the stigmatic area, we have a lot of Normopolis pollen grains.
I'm curious because Normopolis are so diverse that I always thought it would be good to check this diversity with in situ pollen. That is, I mean, how this diversity of dispersed pollen types correspond to real biological diversity.
I found, Natalia, I found the Normopolis in that Coniacian flora and are the same. I found that Normopolis specimens in situ and spread in the sediment. Soon last week, Jürg Schönberger from Vienna told me that one student is interested in studying the specimens. The material is now in Austria, and I hope she could study that pollens in more detail. And later, perhaps I can present another talk here for you about this. I don't know.
Yeah, it would be interesting in light of the enormous diversity and enormous number of dispersed pollen types of this group.
Yes, and there are several types of Normopolis, but this Normopolis I feel that these Normopolis from the Coniacian are different from the Normopolis described by Else Marie from the Campanian Maastrichtian of Myra.
Thank you. Well, do we have more questions, or this was the last one? I think no more questions. So thank you, Mario.
Oh, not at all, Natalia. I'm sorry. I'm really sorry for the the long talk.
It was
1
great.
hour and a half. But I tried to do my best, but it was impossible to show you these things in half an hour. As I told you before.
Thank you everybody for joining us, and see you.
Okay, bye bye.